And our first webinar this year is on clinical lactation studies, looking at design considerations from collecting samples with Dr. Tina Chambers, and also from the analysis standpoint with Dr. Jeremiah Momper, who are part of our MPRINT Center for Excellence at Therapeutics at the Skaggs School of Pharmacy and Pharmaceutical Science and Department of Pediatrics at UCSD. So without further ado, I will turn it over to Drs. Chambers and Momper to discuss some aspects of clinical lactation studies with us.
Thanks so much, Dr. Quinney. So as was stated, we're going to give kind of an overview of clinical lactation studies over the next 40 minutes or so, and then have some opportunity for Q&A at the end if there are questions. So the first thing, of course, in a lactation study, as in any study, is identifying what the research question is. And the primary research question in lactation studies regarding therapeutics primarily is, can the drug be safely used in lactation with respect to the developing infant or toddler who is being breastfed? And another research question that has come up occasionally might be if there is a concern about the safety of this drug when used in lactation, theoretical or concrete, and the drug needs to be taken, how long is it necessary for that breastfeeding mother or lactating mother to wait after the last dose of the drug so that she can resume breastfeeding if that's what she wishes to do? So that could be a research question that has a little bit different focus. And Dr. Momper will be giving an example of one of those types of studies later. So I'm going to spend next few minutes talking about sort of the guidance from the FDA for industry on doing lactation studies. And as stated, there was initial guidance back in the early 2000s, and then draft guidance updated in 2019. I think it's working its way towards final guidance, but this is an emerging area. As we can all appreciate, in the area of safety of medications and lactation, where we have a huge paucity of data, the first and often the only question that's being answered is the drug present? And if so, in what quantity? And frequently then that's results in a milk-only study that addresses that question as opposed to additional questions that might be of interest. So again, the FDA draft guidance, last issued a few years ago, is intended to inform the product label so that studies that are done in response to post-marketing commitments or requirements or otherwise meet certain criteria so that this information can be included in what was updated in 2015 version of the product label, where a section is set aside for lactation safety data. So this is just a cover page, and this is, easily accessible and readable. I'm just pulling out some excerpts over the next few slides to go over in summary what was stated in this guidance. So, of course, the background is that, there have been efforts to improve quantity and quality of information and labeling for drug use during lactation. But when it comes down to it, you can't put something in the label if there aren't data to put in the label. So more interest in recent years and an increase in the number of post-marketing commitments or requirements being issued by the FDA for new products or products with new indications requiring those lactation studies. In the current environment, if there are no data, lactating women and their healthcare providers have to make decisions about drug treatment, initiating it, or continuation of breastfeeding during therapy, where quality human data is not available in the labeling or anywhere. And so for that decision to be evidence-based, lactating women and healthcare providers need the information. So that's the rationale for trying to support good quality data from clinical lactation studies that can be included in the label. And as stated here, the FDA is interested in evaluating the safety of the drug when used during lactation, but also information on how you might recommend minimizing infant exposure if it's appropriate. So several sections of the guidance cover various approaches. In this section, the lactating woman is the subject of the study, and this is what we're calling milk-only studies. So a milk-only study, as described earlier, can be used to detect the presence of the drug in breast milk, to quantify or estimate the total amount of drug transferred into breast milk when plasma concentrations in the mom are known, and to evaluate the effects of a drug on milk production when milk production in lactating women not taking the drug is known, so you have a comparison. And if the concentration of the drug in breast milk is found to be clinically relevant, this finding could lead to further studies. And Dr. Mopper will talk a little bit more about this, but a typical sort of benchmark is if concentration in breast milk is at the less than 10% level, where clinicians or researchers are less concerned about it than if it was higher than that. As stated in this guidance, in general, FDA recommends milk-only studies unless there is a reason to conduct another type of clinical lactation study. So at least it's a first step. And of course, if there's no drug in the milk and it can be clearly demonstrated in a way that's reliable and valid, that there's no drug in the milk, no need to go further. There can be mother-infant pair study designs that include assessment of drug concentrations in infants. So that would be the next step that can provide information on the absorption of drugs in infants through breast milk, assuming there is a drug in the milk, and can include safety assessments in infants enrolled in these studies. And as recommended by the FDA, a sponsor should consider this design if information is already available about the extent of drug transfer into breast milk, and including, and especially if there's evidence that the drug accumulates in breast milk and if the drug is likely to be absorbed by the breastfed infant. There can be variations on the study design with respect to the duration and the dose. So there is a single dose design for drugs that are given acutely. For example, could be a single dose drug, or drugs that do not accumulate with chronic dosing. And in those circumstances, a single dose study may be sufficient. And there could be longitudinal designs for drugs that are administered chronically, given for several treatment cycles. You might consider longitudinal study design, and under that design, samples would be obtained at different time points over that time course. Covariates and other aspects of the lactating mother and infant need to be attended to. So as noted here, maternal weight, maternal age, gestational age of delivery, the stage of lactation, is the mom producing mature milk, length of time postpartum, whether she has other exposures, including tobacco and alcohol intake, other drugs, race, ethnicity, and underlying medical conditions should be collected and reported as part of a lactation study. And important to specify whether the mother is exclusively breastfeeding as opposed to supplementing with infant formula, in particular in determining infant dose. The FDA recommends that studies include only women who exclusively breastfeed. However, that is not entirely feasible under some circumstances because women who are supplementing with infant formula actually represents, in many cases, real-world data and may allow for easier collection of milk that would otherwise be discarded. But to address this, study should report the extent of use of infant formula in participants. For milk sampling, the type of milk collected is recommended. So the study design should specify the type of milk. For example, differences in the composition of foremilk versus hind milk should be accounted for with some drugs because transfer could be affected by the composition of the milk in terms of its the water content and fat content. And the milk sampling method. So in general, FDA recommends collection of the entire milk volume from both breasts over 24 hours. Sampling should occur when drug exposure is at steady state during chronic maternal dosing. And if the dosing intervals are more than 24 hours, then consideration should be made to collect milk over the entire dosing interval or to collect 24-hour samples during the expected time to peak plasma concentration. And the sampling schedule should also take into consideration known PK parameters for drugs with longer dosing intervals. So I'll give you a couple of examples in the next few minutes. The first one is a published study. This study was actually conducted back in 2014, 2015 by the manufacturer of certolizumab pegol called the CRADLE study. This was not to meet a post-marketing commitment. It was a voluntary industry-sponsored study and is an example of a milk-only study design with the goal of including the data in the product label. And the results of the study were published in 2017 in the Annals of Rheumatic Disease. A subsequent additional bit of data on the infants was collected, but to my knowledge was not published. So just to give you an idea on the methods, this CRADLE study was a PK study of lactating mothers receiving certolizumab. They had to have received at least 3 certolizumab doses, so they couldn't be starting on their first dose when they enrolled in the study. They collected milk samples across 1 dosing period. So this drug is given periodically. It can be, in this scenario, can be given every 2 weeks or every 4 weeks. And so if the mom was taking the drug as she normally would every 14 days, then the samples were collected over that period. And if she was taking it every month, then the samples were collected over the month-long period. And they used analytical methods that were developed specifically for certolizumab and the polyethylene glycol component of the breast milk. Average daily infant dose and relative infant dose were estimated, and they also captured safety events, adverse events in the mothers and the infants. So eligibility to be in this study were that the child had to be at least 6 weeks postpartum, but they could be any age at the time of enrollment. So the 6 weeks, allowed the mother to have already established breastfeeding and to have mature milk being produced. But they had a pretty wide age range. So they ended up having infants in the study who were between about 1.5 to almost 17 months of age. They excluded preterm infants from the study, which was a limitation, but reasons to think that there might be differential, at least infant absorption of any drug that was present if the infant was preterm. The mothers could be supplementing with formula, and they captured that information. And they, in order to not kind of muddy the interpretation, they couldn't be treated with any other biologic medication during the time that they were doing the sample collection. They did this at 6 sites in the US and Europe, and they used a protocol that involved home health nurses who went out to the home of the participating mothers and collected the milk. So the study provided an electric pump, and they collected a full expression from both breasts, as was recommended in the guidance now. The milk samples from both breasts were mixed, and they poured off 5 milliliters, which was then sent on dry ice from wherever it was in Europe or the US to a single vendor in the US where the analyses were done. And they collected samples across a single dosing period. So if the mother was taking the drug every 2 weeks, they collected a sample at 0 days, 2 days, 4 days, 6 days, and then every 2 days up to the 14th day. And if the mother was taking it once a month, they collected that same every 2-day sample, and then an additional sample at the 28th day. So the results were that they screened 19 mothers. They had 17 who entered the sampling period, 16 who were taking the drug every 2 weeks, once a month. 56% of the 137 samples that were collected had no measurable certolizumab. For the 17 mothers, all samples were below the lower limit of quantification. They estimated the average daily infant dose was 0 to 0.0104 milligrams per kilograms per day, and that the median relative infant dose was 0.15%. The PEG was undetectable in almost all samples and could not be determined in the 3 remaining samples. And the safety profile, they actually didn't have an internal control group, but the safety profile, they said, was consistent with that of unexposed similar age infants. And so they concluded when quantifiable certolizumab concentrations were less than 3 times the lower limit of quantification, so less than 1% plasma concentration observed with therapeutic doses. this ended up in the product label, as was the goal. And so, as you know, in the product label, Section 8.2 now includes a risk summary for lactation. And this is how the study was translated. So in a multi-center clinical study of 17 lactating women treated with Cimzia at 200 milligrams every 2 weeks or 400 milligrams every 4 weeks, minimal certolizumab pegol concentrations were observed in breast milk. No serious adverse reactions were noted in the 17 infants in the study. There were no data collected on the effects of milk production. In a separate study, and actually this is the study that I don't think is published, but must have been submitted to the FDA, the certolizumab pegol concentrations were not detected in the plasma of 9 breastfed infants at 4 weeks postpartum. And these 9 infants in the data that accompanies this in the label were being varying levels of exclusively breastfed or in combination with formula. And so the bottom line in the label was developmental and health benefits of breastfeeding should be considered along with the mother's clinical need for Cimzia and any potential adverse effects on the breastfed infant or from the underlying maternal condition. So kind of the not very conclusive statements that are oftentimes included in the label. The second example is another milk-only study that we're doing here. It's not completed yet, but this was designed to meet a post-marketing requirement started here at UCSD in the last couple of years. The drug is prucalopride. It's used in a single daily dose for treatment of functional constipation. And the study is being conducted by our UC San Diego Mommy's Milk Human Milk Research Biorepository. It's been in existence for about 10 years. So within this infrastructure, we're recruiting women throughout US and Canada who are currently already being treated with prucalopride and are breastfeeding and willing to take part in the study. So eligibility, the child has to be between the ages of 10 days and 11 months. So mom producing mature milk. And the 11 months was chosen as the upper end of the range because we're asking for growth and development follow-up information through 12 months and 30 days. The mother needed to be treated for at least 5 consecutive days before sample collection started and had to be exclusively or almost exclusively breastfeeding. And different than the study I described earlier, this is a self-home collection protocol that we've used in the Mommy's Milk Human Biorepository successfully over the last 10 years. So the mothers are given instruction to and procedures on how to collect the samples over a 24-hour period. And in this scenario, we asked them to do it from a single breast so that they can continue to breastfeed the baby from the other breast. The dosing schedule was pre-dose within 1 hour before they take their daily dose of prucalopride, and then 1, 2, 4, 8, 12, and 24 hours post-dose. We required that they used an electric breast pump, but didn't provide it for them. And then we captured the maternal and infant characteristics, as I described in the guidance by questionnaire, and then followed up on those infants with neurodevelopmental screening at about 12 months of age, growth, and infant adverse events. So the analysis for this involves, of course, PK, dose normalized area under the curve in milk, dose normalized daily infant dose and % of maternal dose estimates, and relative infant dose estimates. And for this study, the sample size was set at an N of 12. You'll note that the previous study and this one both have very small sample sizes, kind of no power calculation done for these. These are somewhat descriptive. The previous paper suggested that the FDA guidance was they have a minimum of 8 participants. I'm not sure where that came from. We set it at 12 as likely feasible, and that we would do an interim evaluation to look at inter-individual variation after 6 participants were enrolled to determine whether or not we needed to adjust the sample size. So I'm going to stop sharing and hand this over to Dr. Momper, who is going to tell you why milk-only studies aren't the only way to go.
Okay, thanks, Dr. Chambers. Let me share my screen and put this into presentation mode. Okay, so thank you for that. And we thought what would be useful is to shift gears a bit and talk about some real-world studies In other words, acquiring data or evidence generation in more typical scenarios. A lot of what Dr. Chambers discussed were studies that would be conducted in the context of drug development, or at least involve some pharma participation where there's an intent to have data go in the labeling. So the bar is quite high for that. And one of the criticisms of the FDA guidance has been that those studies as proposed in that guidance are difficult to conduct and in some cases may not be feasible. Incidentally, that's still a draft guidance, so would certainly encourage everyone to read it. And if you have comments, you can provide those to FDA for consideration. So like I said, I'll talk about a few examples of studies that we've been involved in my lab, largely in collaboration with Dr. Chambers, to generate some real world data to inform drug use in lactating people and potential infant exposures. So these are often meant to be complementary to full lactation studies. Again, as Dr. Chambers described, in an ideal circumstance, we would collect milk expressed over an entire dosing interval in order to be able to show really conclusively the drug that the breastfeeding infant is exposed to. But often that's really challenging to do. So there's some more novel, creative ways that we can get these data, and I'll describe some of those. The goal is similar. We're looking at identifying exposures of concern or interest, and I would also add, in some cases, responding to emerging needs. And I'll talk about a few examples as I go through this section, where we've been involved in trying to get data out as quickly as we can, again, to respond to things that are emerging from a public health point of view. Some of the challenges in these types of studies that we've seen and have previously been described are lack of awareness among pregnant and lactating persons, as well as their providers, to enroll participants, and then a lack of diversity in these types of clinical cohorts. And there is a need for improving collaboration, and this is true across different data sources, networks, platforms. And so I think this is, one of the benefits of MPRINT is bringing different people together who work in different networks and sharing a common goal towards improving medication use during pregnancy as well as during lactation. So I'll give one example. Again, this is fairly recent that we've been involved with in the past couple of years, but it really focuses on antiretrovirals in milk. So as many of you likely know, antiretroviral treatment is recommended for pregnant and lactating individuals. And this is, of course, for the primary treatment of HIV in the mom, but also preventing perinatal HIV transmission to the fetus or to the breastfeeding baby. So it follows as well that all babies born to people with HIV should receive HIV medications as soon as possible after birth. And these statements are taken from the perinatal HIV guidelines, and I've provided a link there. So for many years, going back to the 80s and 90s, when antiretroviral treatment was available, the recommendation in the United States was largely to avoid breastfeeding. We've seen a shift in the past couple years, and this is again taken from the NIH panel. This is the recommendations for the use of ARV drugs during pregnancy. But this is in particular looking at infant feeding. So a major shift occurred a few years ago. This is updated in, as you can see, January 31st, 2023. And I've highlighted one important point, stated that individuals with HIV who are on antiretroviral therapy with sustained undetectable viral load and who choose to breastfeed should be supported in this decision. So really moving towards a model of shared decision-making. And this is largely in recognition of the vast benefits of breastfeeding, both to the mom and to the breastfeeding infant, and that the risk of transmission of HIV in suppressed individuals with HIV transmitting to the infant is quite low, actually, if the mom is suppressed. It's not zero, but it is quite low. So again, this is a big sea change where now there's a push to support women who choose to breastfeed, and that allows, or at least opens the door to a lot of considerations about antiretrovirals in milk, whereas previously it was not really thought of that much because, again, the recommendation was simply not to breastfeed. So giving a few examples here of why this is important and why we need to think about antiretrovirals in human milk. So there is a variety of concerns that come to mind here. The first is the potential for the development of HIV resistance in breastfeeding infants who go on to acquire HIV. So of course, we hope that any infant born to a mom with HIV doesn't go on to acquire HIV. It's rare that it happens, but unfortunately, it does still happen. And so the concern, number one bullet point here would be that if an infant acquires HIV and they're exposed to low levels of antiretrovirals through milk and they absorb those, that could promote HIV resistance. In other words, the infant could develop a resistant strain of the virus that could be resistant to individual drugs or potentially entire drug classes. So that can be, catastrophic for a young person having to go their entire life with antiretroviral therapy when some of the most potent and widely available drugs are not effective. The second is managing dosing requirements in infants who receive the same antiretroviral drug to prevent perinatal transmission. So as I mentioned, all infants born to moms with HIV should receive antiretroviral drugs. And so therefore we have the balancing of these dosing requirements where the infant may be acquiring some drug through breast milk, separately acquiring drug that's dosed directly to the infant. And we need to think about how those dosing requirements may change in that setting. And then thirdly would be any potential long-term toxicity. These are, of course, drugs that mom takes chronically. And so there is the potential for a breastfeeding infant to be exposed from birth all the way to 1 year of age or whenever they stop breastfeeding. So in interpreting drug concentrations in milk, we're often looking at relative infant doses or RIDs. And this is simply an estimate of the daily infant dose expressed in milligrams per kilogram that's calculated based upon an average concentration in milk. Here, that's milligrams per mil, as you can see. And then, let me see if I can get a laser pointer. And then multiplied by a typical milk intake, which is 150 milliliters per kilogram per day. So this gives a daily infant dosage. And then we can transform that into an RID or relative infant dose by looking at that daily infant dose on the numerator divided by the daily maternal dose, notably in terms of body weight, so milligrams per kilogram, and then this is often expressed as a percentage. So as Dr. Chambers mentioned, there are WHO thresholds. These have been around for a number of years. They're somewhat arbitrary, but the acceptable limit is less than 10%. Caution is 10 to 25, and then quote unquote unacceptable is above 25%. Again, this is for the relative infant dose. So an important question here is whether these benchmarks are reasonable or appropriate for antiretroviral drugs. Again, I would look at this through the lens of the potential for the development of resistance in the breastfeeding infant who goes on to acquire HIV. So I'll give some examples and some context around this. At the time when these recommendations were changed in the United States, and incidentally, these recommendations for breastfeeding and HIV are largely changing across the world now, there's not a lot of data. This is one particular data set from Switzerland. So this is the Swiss mother and child HIV cohort study. And you can see that the relative infant dose for a variety of commonly used antiretroviral drugs across the board is less than 5%. Some are extremely low, like rilpivirine, for example, 0.02% and so forth. But I'd also point out here that the ends, that the number of participants that contribute to these types of data are extremely small. So in some cases, we're making assumptions based upon 2 participants or 2 milk samples collected from 2 unique individuals. So that's a kind of a major liability in terms of our current understanding of where things are with antiretroviral drugs in breastfeeding. So I'll talk about one study that we were involved in to generate additional data, and I'll highlight in particular dolutegravir, which is an integrase inhibitor. It's one of the most widely used antiretroviral drugs, widely recommended in guidelines both in the US and internationally. This was actually kind of an add-on study. The primary study, which is Impact 2010, also called VESTED, which I was a pharmacologist for, this was an outcome study that looked at efavirenz-containing regimens relative to dolutegravir-containing regimens. So really looking at virologic outcomes during pregnancy. But we also had an opportunity to extend this study in a kind of a secondary aim to look at milk exposures, and in a unique way, we looked at time-matched maternal plasma, breast milk, and infant plasma. So we took samples at 6 weeks postpartum. Again, the kind of triad of maternal plasma, breast milk, and infant plasma all collected at the same time. And we were able to get samples from nearly 200 women. So showing some of this now. So this is, again, 200, approximately 200, 192 individual participants, including their infants. So on the left-hand side is simply the maternal plasma concentrations. The shaded area represents the interquartile range for historical non-pregnant data. Again, these would be week 6 postpartum maternal plasma concentrations. I'm just highlighting DTG or dolutegravir here. And then we also collected breast milk and infant plasma, similarly 192 for each. So just focusing in on the breast milk and infant plasma data, the median with interquartile range DTG concentrations in milk were 67, 91 was the median with an interquartile range of 67 to 123. And then looking at infant plasma, we were at 69 nanograms per mil. So clearly the drug is in breast milk, it's absorbed by the breastfeeding infant, and we can measure that in the systemic plasma of the breastfeeding infant. These concentrations indicate a relative infant dose less than 5%, so certainly not extremely high relative to the dose that the mom is getting. But if we look a little bit further into this and consider some of the PK and PD characteristics of dolutegravir, there is potential for concern. So the in vitro 90% protein-adjusted MIC, or IC90, for DTG is 64 nanograms per mil. So it's actually remarkably close to the median concentration that we're seeing in infant plasma. But of course, there's a lot of variability around this estimate. The interquartile range 41 to 95, and of course, you're going to have at the extreme end a lot of infants who are well below the 69 nanograms per mil. So this does, again, paint a picture where there is a cause for concern for the development of resistance. If any of these infants would go on to acquire HIV, it's unlikely that this low exposure to dolutegravir is going to be therapeutic, meaning preventing HIV replication. But on the other hand, these low levels of DTG that are hovering around the IC90 may promote the development of resistant virus. Again, that can really be problematic for the infant, such that for the rest of his or her life, they unlikely to be effective receiving commonly used antiretrovirals, including of which dolutegravir is one. Another interesting finding from this study is that concentrations of dolutegravir in infant plasma are about 5 times higher than we would have predicted based upon the DTG dose through breast milk. So if we look at the breast milk concentrations and assume a range of commonly or common milk intake in the breastfeeding infant, these concentrations in infant plasma are quite a bit higher than we would have expected, which does suggest that actually bioavailability in milk for this drug may be higher than some well-characterized commercial formulations. Those include liquid oral suspensions and dispersible tablets. Separately, we're working with the Impact Network and ViiV GSK on a PK study of DTG in newborns who are actually dosed directly with these formulations. But again, an interesting finding that the bioavailability appears to be different in milk as compared to commercial formulations. And the other thing, as I mentioned before, is that when we think about then dosing dolutegravir in these babies, we have to think about the additive effects of milk or concentrations that are acquired through milk, as well as the drug that's dosed directly to the infant. It gets even more complex when you think about the first few days of life, because then you have drug that's acquired transplacentally. On the left-hand side here, we're showing infants born to mothers receiving DTG. These are the first few days of life, and you can see that all infants have detectable or measurable dolutegravir concentrations, which can be pretty high, 3 microgram per mil or higher. So this is really a challenge here, because we have transplacentally acquired drug, we have drug that's coming from breast milk, and then, like I said previously, there's a lot of interest in now developing dosing for dolutegravir from birth. So a lot of moving parts here, but just kind of highlighting the utility of, in this case, a study that was added on to an existing larger trial that looked at outcomes. We saw an opportunity to collect additional data to inform information about medication use during breastfeeding for these drugs, and that was added on at a week 6 postpartum visit. So I'll give 2 other examples of areas that we've been involved in recent years and, kind of identifying emerging public health issues as it relates to drugs in milk. So this deals with remdesivir and its metabolite in breast milk. So you likely recall there was a lot of confusion or mixed messaging around breastfeeding during the early days of the pandemic. But eventually the recommendations from the CDC, which are quoted here, are that when possible, breastfeeding mothers with suspected or confirmed COVID-19 should continue to breastfeed while taking hygiene precautions. So this raises some questions about drugs that are used for COVID-19, and remdesivir was the first drug that was approved by the FDA for the treatment of COVID-19. Remdesivir is a nucleotide analog. It inhibits RNA polymerase, and we acted pretty quickly. We worked with Gilead. They did have a post-marketing requirement to do this, but again, we tried to turn things around quickly. And in doing so, we leveraged samples that were in the Mommy's Milk biorepository at UC San Diego that Dr. Chambers directs. We also acquired a few samples from a PK study, Impact 2032. This was a PK study in pregnancy for remdesivir, but we were able to collect a few breast milk samples added onto the study. So again, the key point here that I'm threading through is that we need a lot of creativity often to collect these types of data, and when possible, thinking about existing studies and ways that we can acquire breast milk data can often be quite helpful. So for remdesivir, shown in the orange, we did show that all remdesivir samples were below the quantitation limit. The assay here was 100 nanograms per mil. And then we also looked at the major circulating metabolite of remdesivir, which is this GS441524. The majority of those samples were also below the quantitation limit. Some were measurable, and if we just look at those measurable samples, the concentrations were about 152 nanograms per mil. So quite low. This is probably a situation where the ultimate conclusion is there's really no reasonable risk to the infant based upon these low concentrations that we see in milk. These data are, to my knowledge, in the label, although again, this was not a full lactation study. It was really leveraging existing biospecimens, both from a clinical trial as well as the biorepository, to respond quickly and provide some information as things were evolving in the pandemic. And the last example I'll give is related to CBD, or cannabidiol. So as all of you likely know, in the last 5 or 10 years, we've seen an explosion in the use of cannabinoids in the United States. Even states that don't allow recreational use of cannabis, often CBD is readily available. And we've seen rampant CBD use in different scenarios, including in pregnancy and postpartum in lactating people. So this was a study that looked at leveraging existing data, again, using the biorepository at UCSD. We obtained 200 milk samples from 181 unique lactating women who had self-reported CBD use. And those concentrations are shown here, as well as the time after dose on the x-axis. We looked at the distribution of these concentrations depending upon the use method for CBD in the individuals. That would include a joint, blunt, or edible, or oil or pipe, which tends to lead to higher exposures in maternal plasma and subsequently in breast milk. And so this, in terms of the doses administered to virtual infants, and I'll describe some of that modeling in just a bit, are going to be higher based upon ingestion through a joint or blunt or edible or oil or pipe. So putting this all together, we had worked with a group at the University of Waterloo in Ontario, Andrea Edgington, and at the time, her postdoc, Cindy Yoon, to look at PBPK modeling of CBD exposures based upon those concentrations that we showed previously. So this was a PBPK model in infants. The dosing effectively was a function of the maternal intake of CBD. Shown on the left-hand side at 5 milligrams per kilogram is the CBD exposures that come from Epidiolex. So Epidiolex is an FDA-approved formulation of CBD that's used for the treatment of certain types of childhood seizures. So we use this as a benchmark here, the 5 milligram per kilogram dose. And then the predicted plasma AUCs based on postnatal age of the breastfeeding infant from 0 to 7 days out to 1 year of age. And as you would expect, based upon what I showed previously, our predictions are such that if the mom is using an oil or pipe, that does lead to higher exposures maternally and subsequently higher exposures in the breastfeeding infant. These are all well below the exposures in, again, coming from Epidiolex at a much higher dose. The conclusion here is not that these are safe and safe for the infant, but rather, again, just showing a kind of benchmark here as it relates to Epidiolex. So a few high-level thoughts about how we can improve interpretability and generalizability of lactation studies. So the first is trial design, really thinking about consistent sampling should be reproducible. When possible, it's useful to, as I showed with the dolutegravir study, to get maternal plasma, breast milk, and infant plasma. And often there's some things that come up here that are unexpected, like I showed with dolutegravir, the bioavailability appears to be higher in breast milk, such that those concentrations in infants are higher than we would have expected. Collecting across several time points after birth is useful, and then mother-infant pairs followed longitudinally. And it's really important to have a good dosing history in the mom, that is. Without having good history of maternal drug intake relative to sampling, our hands are often tied in terms of conclusions that we can draw from data. There are also a variety of laboratory considerations that need to be considered. These include appropriate assay validation. So this gets a little tricky because ideally we really want to develop and validate the assay in donated breast milk. It shouldn't be an assay that's simply ported over from serum or plasma or whole blood. It really should be from the ground up, done in breast milk. Breast milk is a relatively heterogeneous matrix in terms of lipid content, protein content, et cetera. So it's a little bit different than blood or plasma from that standpoint. So there's some considerations there. The milk fraction really should be standardized. Generally, the recommendation is to use whole milk. There should be a very clear justification for use of skimmed milk fraction, which is generally, again, not recommended. Storage conditions are also very important to consider in terms of validating a particular method, and notably would be stability. So, for example, if we have milk samples that are collected at home from participants and then go to a biobank, we need to think about the conditions in which that sample is encountering. So is it going to be at room temperature for a number of hours? Is it in the refrigerator or the freezer at home? When does it reach -80 in a research setting? All of those things can affect stability, and then also thinking about long-term stability. So if the sample sits in a freezer for 2 years before it's assayed, we really need to have 2-year stability data in milk along with it to show that the drug is stable, or at least account for degradation over time. And those types of stability studies can't really be sped up. So if you don't at the front end think of this and put spiked QC samples, for example, in the freezer and keep those for several years, you can't go backwards and estimate stability. So that's something that needs to be thought about on the front end. And then also milk composition. So the lipid and protein content, like I mentioned, it's good to measure those and, when possible, correlate with drug concentrations. We have seen some drugs have huge changes as milk composition changes. The most notable one that I've seen is delta-9 THC, which is extremely lipophilic. When you have more fat-heavy milk, the THC concentrations are several fold higher than milk that has lower fat content. So to wrap up, some conclusions across Dr. Chambers and I, our two sections. First will be clinical lactation studies should be fit for purpose, depending on the drug and the research question. Information from single-dose studies and milk-only studies is often quite limited, and so thinking about ways to leverage additional samples, whether they be maternal plasma, infant plasma, et cetera. And lastly, reliability and validity of lactation studies can be influenced by the design, collecting samples at a variety of time points after birth, and then several different considerations for laboratory and storage conditions. So I'll wrap up here, and I think we can open it up for questions.
Thank you to both Dr. Chambers and Dr. Montfort for some very interesting content on study design and lactation from the two different perspectives they've given. So at this point, we'll open for content or for questions. Feel free to put something in the question and answer box or raise your hand and we can promote you to ask your question or provide comments as well. So while we're waiting, I can start. One of the comments Dr. Montfort made when the discussion was that the bioavailability can be higher in milk for some drugs. Is this typical? Do we know what the mechanism of that is compared to a typical formulation, or is there a specific type of drug that would be more likely to have higher bioavailability in milk?
That's a good question. I don't know offhand. I think it's, probably if you looked at BCS classes and some of the permeability characteristics, et cetera, there may be some trends there, but it's not something I've explored in depth. Some of it may also be the developing infant gut and the bioavailability that we think we know in infants is often kind of scaled from adults. And so there's some assumptions around bioavailability, even irrespective of what we see with drug through breast milk. So I think it's an area that's evolving, and I'm sure we'll learn more in the coming years.
Yeah, from your standpoint on both of the approaches, what's the most difficult component of doing these studies in breast milk lactation?
Well, I'll say, as with any studies, especially if it's a rare exposure, the difficulty is locating the people who've had the exposure. If you're looking for somebody who's interested in participating in a lactation study, and then the motivation to do sample collection that requires that they not just send you one sample, that they do it over a time period. Once enrolled, I think we get good compliance and we have good retention, and moms who participate follow instructions and are really interested in the outcomes. But it's finding the needle in the haystack if it's a not extremely common exposure.
Yeah, I agree. I don't have much to add. I think, as I alluded to when I gave my kind of introductory comments, I think having samples that are representative of the patient population as a whole has been a challenge. I think, sometimes it's a little bit skewed in terms of the people who are donating samples. And so we ideally would like to get a more diverse representation in these types of biobanks. From the analytical side, I think the issues around method development and stability are important to think of early, because often if you miss that when you're getting a study started, there's not an easy way to go back and correct for it.
And one of the things Dr. Montfort talked about was the importance of having sample times after dosing, or dosing times and sampling. And I know, Dr. Chambers, in a lot of your studies, you're using, samples that are collected as part of a standard of or at home and standard of care. And how are you finding that you're able to collect the accurate dosing times that you need for those, or what's the impact of that on your outcomes?
Yeah, so we're the trade-off here with the home collection is ease of being able to enroll people wherever they are in the US or Canada, and then relying on the mom to do what we're asking her to do. So it's not under observation. So different than doing it in a clinical setting, or like the study I described where the home health nurse is going out and actually observing the collection and capturing information. So we're relying on maternal report, but capturing is detailed information about the timing of exposure dosing relative to the capturing of the milk sample. A little bit beyond that point, we have had success that we didn't expect. We didn't know what the result would be in asking mothers to self-collect both infant and maternal plasma using a Tasso device to pair with the milk samples. So all good on that end, but we do have the sample collection variability, which we don't observe. So they're instructed, to put it in the refrigerator and to send it overnight on ice packs. And then we aliquot it and put it in the freezer. And as Dr. Momper says, it can sit in the freezer for some period of time. So we have all of those, question marks about what happens in terms of stability and the variability of how the samples are handled. We are doing, I think, as you know, Sarah, comparison study to clinic collection versus mimicking home collection standards to see, are there differences between what we get in terms of the assays for specific drugs in those milk samples, splitting the sample and treating it differently, and at least looking at doing the assays relatively soon after the sample's collected. We found that there are no differences except for drugs that we think are impacted by the amount of time they sit out on the counter. So impacted by light, for example. So that's positive. I'll just veering off here, I'm curious whether Jeremiah I think this whole storage issue is so important. And the fact that, as you say, it's, completely reasonable to think over the course of a study that you would have samples sit in the freezer for a year or 2 years before you would do the assay. And in the case where there are rare exposures, you got to build into the study design. But I'm curious whether you think there would be some value in going back in the biorepository where we have exposures that, whatever say it's a SSRI, for example, where we collected the sample from a mom 10 years ago and it's been sitting in the freezer for 10 years, same drug, different mom, 5 years, same drug, different mom, 1 year. Would there be some value in comparing those, even though it's not same sample stored different at varying lengths of time?
I think potentially yes, although you may have had to measure it at some point, so you have kind of a baseline to compare it to. And I think the storage question is not only long-term storage or storage at different temperatures, but also things like freeze thaw, where it may be in the freezer, out of the freezer 3 or 4 times before you actually measure a particular analyte concentration. So I think it's complicated, and my take-home message is, include these things early in a study design. That way, you have it and doesn't come back to bite you, because we want these data to be as robust as possible and inform clinical decision making, make it into labeling, things like that. And it'd be unfortunate, and I have seen where studies fall apart because the analytical data are not very good.
Thanks.
Okay, I'm not seeing any other questions. So at this point, I'd like to thank Tina and Jeremiah for joining us today. And we look forward to hearing more about your work in milk clinical studies and learning about that. We also invite everyone to join us for next month's webinar to address the effects of, or learn about how we can better use artificial intelligence and AI to help with our research. So we look forward to seeing you then. Thank you.
Thank you.
Thank you.